So since Restasis is approved, 20 years passed, we see quite a number of new treatments for dry eye. So what are the unmet medical needs? Dry eye is a symptom-driven disease. So patient want something work fast, work well. Something that make their eyes feel good, see better, and convenient to use. Doctors want something that really, ideally, addresses the underlying mechanism of the disease in dry eye, like repairing the ocular surface, restoring the integrity, and the homeostasis. So, do we know how to do that? Not really. So at Seinda, we go straight to tear biomarker research to first understand what's going on in the disease from real patients. We collect lots of samples, do lots of research from real dry eye patients to uncover key mechanisms and targets to help us to develop diagnostics and therapeutics. So our lead dry eye treatment has achieved proof of concept in the United States phase 2 study. Now we are here to raise funding to support phase 3. So we get our first VC funding for Seinda Pharma in 2020. And now we are looking into end of phase 2 meeting with FDA. So how does our drug work? It's a multimodal mechanism of action that really addresses multiple key mechanisms in the ocular surface. In the cornea, we know tight junction breakdown, we know epithelial cells undergo apoptosis. We need to fix that. We know in the conjunctiva, it loses goblet cells, mucins, so tear film stability go down, patient have symptoms because lose the lubricating effect. We have to fix that. And we also know epithelial cells, in order to make them happy, they need lots of energy, lots of nutrient, because they are highly active cell types. They turn over every 5 to 7 days. So here are just some of the data we have. Our drug can induce M2 macrophage, which has immune tolerance property, and it also have tissue repair property. So we saw in dry eye model, M2 go up, and it brings up IL-13, and it restores goblet cell numbers, and its function and mucin production. And we also see our drug brings down inflammatory IL-1 and other cytokines. Another way this drug works, inhibits epithelial cell apoptosis, and it neutralizes oxidative damages. So here is our proof of concept study, phase 2 trial in the United States. Because it's a new MOA, the objective of this study is multiple. First, safety, tolerability, of course. And then we want to know, do we have a proof of concept drug? And we also want to know what are the key endpoints, the key inclusion criteria, and the treatment duration, and then, of course, which dose to go forward. So to answer all these questions, we designed a study, 4 arms, 3 different concentrations, and very simple inclusion-exclusion criteria. Only 2 criteria, corneal staining and symptom. And our primary endpoint is corneal staining and dryness. And secondary endpoint, Senti score, conjunctiva, hyperemia, and of course, different regions of the corneal staining. And in this study, we also put in our own quantitative MMP9, and we also collect Schirmer at the beginning and the end, and also artificial tear. artificial tear use. So it's in 10 sites. We enroll 200 patients. And here is their baseline characteristic. On both symptom scores, you can see they are moderate to severe in symptoms. In the corneal staining, they are moderate, while they are really a big range because our inclusion criteria was 5 to 16. So we enroll purposely a full range of dry eye patients in this study. And we have 195 finished the study. And here's our primary and secondary. The top are the corneal staining, the central, and the total and the central, and then the dryness and the Seinda score. So just the top view of it, we see consistently improvement over time in both corneal staining and symptom. And we also see by 8 weeks, all 3 concentrations are showing improvement better than vehicle. May not be statistically significant at this top looking, but surely some of them are. Now, a closer look of different endpoints, because we want to answer the question, do we have a proof of concept? So here are dryness, Seinda, and then the Seinda subscale severity. What we saw was all 3 symptom scores we see consistently over time improvement. And at 8 weeks, we see significant over vehicle in all 3 doses by 8 weeks. But we also noticed very fast action 7 days after treatment. Dose response. Very, very convincing in terms of this drug improves symptom. Now, does it improve corneal staining? Because we have a wide range of staining patients, so we want to answer this question, does it help corneal staining? So we look at the subset with higher corneal staining, and from this graph, you can see, yes, this drug improves corneal staining. If we look at the red line, which is the 0.5, it really pulling away from the vehicle group and continue to improve over time. Another way to look at the data was percentage in each group, because it's a small study. So it's important to look at the percentage of response. Surely you can see all 3 groups, even at 2 weeks after dosing, it's really much higher percentage of patients improve their corneal staining. So with our quantitative MMP9, we also want to look at subset of patients with our own threshold of 31 nanogram per ml. And here are some of the analysis results. This is the 0.5 over vehicle. And in each group is about 20, 21. We see very striking symptom improvement as early as 2 weeks already, had the potential, basically significant difference if we have just a bit more subjects. And another thing that's very striking is the extent of the improvement over vehicle, 17 points over vehicle. And corneal staining, 2 weeks after, already see substantial improvement. So with this drug, what's our positioning and differentiation? It's a novel multimodal mechanism of action, and it really comprehensively improves the ocular surface. So give patients, a wide range of patients, the improvement of the signs and symptoms. And it's twice a day, and it's also patients feel the benefit minutes after, and also substantially improve over time. So we are raising funding for phase 3, and Last but not least, I want to thank our investigators, our devices, and consultants. Thank you.